out. All right, so we are in lecture unit 3. You're going to go all the way down to the unit 3 resources. Come on, Blackboard. And then right here, second from the bottom, week 4 review session. That's where we're at today. So if you'll click on that folder, wait for Blackboard to do its thing. Here is the handout, and then the video, I'll post the link right underneath. So we're going to go through this worksheet today, and we're going to be covering host microbe interactions and nonspecific immunity. But what I'm going to recommend you do, because we've already done the knowledge checks for this, you've had your week 4 activity for this, I'm going to recommend that as we go through this, you can have your notes handy, but try not to use them, okay? Because we don't want to be using our notes as a crutch. We want to make sure we're figuring out what's in our brain and what we kind of need to keep working at to get there and to be able to retrieve. So once you have that handout ready, we're going to go ahead and get started. I'm going to zoom in on this a little bit. Oh, actually, before we start, there's something else I wanted to show you. Okay, so if you go back into, oh, hold on, I'm not sharing the right screen. Here we go. If we're back in the Unit 3 resources folder, if you go up here to the Microbe-Host interactions, if you click on that one, I recently, just this morning, added a folder called flowcharts. Last semester, my students just overwhelmingly, they loved flowcharts. And so I went through all of the microbe-host interactions, and I made like flowcharts for each of the steps. And so you guys did the modes of transmission as part of your week 4 activity. And if that's your jam, if you like them, I have them for all the other steps as well. So they're not for a grade. It's just in case that's how your brain works and you want to study them or use them to study. Okay, now let's go ahead and get started on this. So again, without your notes, I want you to come up with the difference between contamination, infection, and disease. So what's just a general definition for each of those, and then an example. Okay, so I'm going to give you about 30 seconds to just put your thoughts down. Again, try not to use your notes, and then I'm going to talk through it with you. So just take about 30 seconds. Okay, so contamination, keyword here is going to be exposure to a pathogen. Okay, and I'm going to come back and give an example after I define each of them. So you are exposed. This pathogen has accessed your body. Okay. Infection is when there's, if I could type, replication of the pathogen in or on the body. And disease is when it is successfully making you sick. So I'm going to put sick, but basically it means there's a change from what is considered normal. Okay? And don't worry, I'll give an example to help make these make sense. Okay? So think of it, and I think I might have given this example in the video. But a couple years ago, my niece, well, a long time ago at this point, my niece was sick, and I didn't really realize that I had just gotten to see her. I just picked her up from daycare, and she gave me this big old hug and was, telling me all about her life as like a 2.5-year-old. And all of a sudden, I'm like, oh yeah, and my mouth's wide open, and she like full body like sneezed into my face and my mouth, right? So at that moment, I was contaminated. The germs from her nasal cavity and mouth, right, were now on my body. Okay, so I was contaminated at that exact moment. Now, let's pretend I had better luck and my mouth and everything had been closed and she had just like got her respiratory droplets on my cheek. If I had just wiped it away, that would be the end. It would have stopped at contamination because I would have removed them, right? They wouldn't have had a chance to replicate. But because that wasn't the case, right, I went from contamination to infection because all the germs that got into my mouth when I was loving on her, some of them were able to replicate. Okay, so I was then infected. Now, sometimes it'll stop there. And maybe my immune system would have cleared it and I never would have gotten sick. Okay, so it can stop at infection. In my case, it didn't, right? A couple of days later, I woke up and I was also sick. So when my body was no longer normal, I had a disease. Okay, now I know in society, common conversation, you don't go, oh, you have strep throat, that's a crappy disease, right? We normally save the word disease for like big things like diabetes and heart disease. But technically, once your body has deviated from its normal healthy standard, it's a diseased state, okay? All right, do we have any questions on the difference between these? You are definitely going to want to know them come exam time. Okay. Now, if you have any questions, feel free to come off mute or type something in the chat. All right, so now I'm going to want you guys to answer this for me. Identify the mode of transmission for each scenario. Okay, so you need to think back to the different types of transmission. There we go. We have direct transmission, which is like you are coming into contact with that pathogen. We have indirect transmission, where there's kind of like a middleman, like a fomite, an inanimate object that's introducing the pathogen to your body. And then we have vector transmission, okay? So if Tanya ate a chicken that was contaminated with salmonella, what would that be? What is that mode of contamination? I'm sorry, mode of transmission. I don't know why I can't talk today. Does anybody remember? Chicken is food. Oh, sorry, I couldn't hear you.
Oh, sorry, I said direct.
Close. That is going to be indirect because the pathogen is on the chicken, which then is getting into our body when we eat it. So that's going to be indirect. And if we want to be more specific, it's going to be vehicle transmission. Because remember, air, water, soil, food, if the pathogen is attached or associated to with any of those, it's considered vehicle transmission. All right, B, Tracy got pneumonia after a sick person coughed in her face. So in this case, the cough, right, puts the pathogen directly on or in, what's the name, Tracy's body. So what's that one going to be? It's going to be direct, but more specifically, it's going to be respiratory droplets. Because again, when you cough, kind of all that spittle that comes out of your mouth, the pathogen can be attached to it, and then it gets into your body. All right, for C, Joey's dad has a cold sore. His 2-year-old son drank out of the same cup as him and got infected with HSV1. So again, the virus in this case is associated with the cup. And so the son is coming in contact with the pathogen courtesy of the cup. Okay, so this is going to be indirect. And then it's going to be more specifically fomite transmission because it's that cup that was contaminated. And when he came into contact with the cup, he also got contaminated with the virus. And then I don't know why I was a little redundant here, but Stella got strep throat after using the same straw as her sister. So again, the straw is a fomite, okay? So it's the fomite that was contaminated. So this is also indirect fomite. All right. So for number 3, one of the big things people get mixed up on the Unit 3 exam are endotoxins and exotoxins. And you're going to want to know these inside and out. And there's a table that you did in the PowerPoint and in the content videos. So this is just another way of making that. But I want you to go ahead and take about a minute or two, and I need to figure out where that document is so I can add it. Here we go. And go ahead, I'm going to put endotoxins on the left and exotoxins on the right. But I want you to just use your memory at first. Okay, because remember, endotoxins and exotoxins, they're essentially opposite of each other. So whatever you put over here for endotoxins, you're going to put the opposite for the exotoxins. And there's a handful of things you're going to want to remember about them. So just using your brain, go ahead and put down what you remember, and then go back and use your notes to fill in any gaps. I'm going to give you about 2 minutes, and then I'll start going over it with you. Word drives me crazy. There we go. All right, I think it's been about a minute and a half or so. So if you're still working on it, no problemo. We're going to go ahead and start talking about this a little bit. So the big thing, well, there's a couple big things. The first thing is look at the name, okay? We know they're both toxins, right? So we know that the one thing they have in common is that they are little weapons, right? And you can have a better word for that if you want, but they are meant to weaken and harm the host. That is the goal, okay? The other thing you want to remember is that these are made only by bacteria, okay? Viruses do not make toxins, and really that's kind of what we're talking about initially. So right now, in the scope of our conversation, we typically focus on bacteria and viruses. For now, these are bacterial things, okay? There are toxins made by like a fungus and things, but when we get to unit 5, it's less, is it an endotoxin or exotoxin? They can make toxins, but we don't differentiate them in this capacity, okay? So in this class, when you hear me say this is an exotoxin, this is an endotoxin, your brain needs to go, ooh, we're talking about a bacterial pathogen. Okay? Viruses do not make toxins. Okay, so that's kind of what they have in common. Really, they don't have a whole lot, except that they're produced by bacteria in order to weaken and hopefully for them, take over the host. So look at their names. They both have toxin in them. So one of the big differences is the endo versus the exo, right? So endo always means within. I think of it as endo into. They sound kind of similar. So this is made within the bacteria and it stays with the bacteria, okay? And we're going to do the exotoxin after I get this list made. These bullet points are probably gonna mess me up, so let me scooch them over a little bit. All right, so all toxins are made inside of a bacteria, but endotoxins are gonna remain associated with that bacteria, okay? And we've already talked about in Unit 1 a really important endotoxin. That's LPS. That's associated with all gram-negatives. So all gram-negatives come equipped with an endotoxin. That's one reason being infected with gram-negative bacteria can be so cruddy. Because when we take antibiotics to get rid of them, we essentially like blow them up and it releases all those little endotoxins throughout our system. And we feel a little cruddy. All right, the other big thing to remember with endotoxin is that this is a carb protein molecule, okay? It is a carb protein molecule. And so what that means, sorry, I'll try to write in text boxes, gets a little hard. Because it's part carb, part protein, it is not sensitive to heat. Let me rephrase that, not heat sensitive. I'm sorry, y'all, carb lipid molecule. I'm sorry. Carb lipid molecule, lipopolysaccharide. Lipo is lipid, polysaccharide is carb. I apologize for that. So carbs and lipids are not as heat sensitive as proteins. Okay, of course, too much heat can mess up anything. But these, because they're not as sensitive to heat, they tend to cause a fever. Okay, they do cause a fever. They also cause just more general signs and symptoms. A lot of time you get like the malaise, kind of the aches and pains, just like get that general like feeling off. Okay, you're kind of achy, you're kind of tired, you're just like bleh. I know it's real scientific, okay? So these general symptoms, okay? So that's not how you spell symptoms, but we're just gonna go with it. I can't just go with it. I don't know how to ignore typos. That's one of my like little kryptonites. There's a typo, I've gotta deal with it. So that's going to be a big difference between these endotoxins and exotoxins, okay? So because they give the same general signs and symptoms, we say that an endotoxin is not disease specific. So in this class, we're mainly going to talk about LPS. There are other endotoxins, but it doesn't matter which endotoxin it is, it's always going to cause a fever. It's always going to cause malaise. It's always going to cause fatigue. Doesn't matter which bacteria. It doesn't matter which disease. If an endotoxin is responsible, you get these same general vague signs and symptoms. Okay? So those are some of the big ones. Because, excuse me, because they are so general, and right, a fever typically is not very dangerous. It can be, but not typically. These symptoms are just kind of weak compared to things like a neurotoxin. We say they have a high LD50. So I'm going to kind of put that with my little fourth bullet here. They have a high LD50. Remember, LD50, oh, you're welcome, Preeti. Have a good day. Because it's a high LD50, that means it takes a lot of the endotoxin to cause any damage, okay? Remember, it's an inverse relationship. The lower the LD50, the more potent and dangerous it is. So endotoxins, because they just kind of typically cause some vague discomfort, we say they have a high LD50. There's too many of them, it can wreak havoc, but typically not a huge problem, more of like an inconvenience. Okay. Yeah, that's a good way of saying it. Yeah, they're less virulent. So where we kind of have to be careful with that, because I love that you said that. A lot of times when we say something is more or less virulent, we're talking about the pathogen itself. And what makes pathogens more virulent is their virulence factors are more like dangerous. So with botulism, that pathogen that causes botulism is very virulent because its toxin is the most dangerous toxin that we know of. So the toxin has a very low LD50. It takes barely anything to kill somebody. And so it has a very potent virulence factor, making it a very virulent pathogen. So for this class, if like on an exam, you said, hey, this has a low LD50, so it's very virulent, like yes, like you're on the right track. But technically, we tend to say something is more or less virulent when we're comparing two pathogens. But you're definitely on the right track with that. Okay, so let's go ahead because I know we have a lot more for this stuff and non-specific immunity to get through. It was very ambitious trying to get through two big topics in an hour. I don't know why I just did that. So we're going to go ahead and compare them to exotoxins now, but y'all, they're pretty much like the exact opposite. So if you remember one, you're going to be good because you'll by default know the other type of toxin. So exo means exit. So this gets made inside the bacteria, but then the bacteria releases it into the host. Because if you look on the PowerPoint, and I sent out a little note about this, let me actually pull the PowerPoints up for y'all real quick to really make my point here. In this PowerPoint, right, there are lots of examples. Let me see, where are some? Like right here, we've got all these diseases that are associated with some of these. You don't need to memorize all these details right now, okay? You need to know what ID50 is. You need to know what parenteral transmission is, but you don't need to memorize all these diseases. And the reason that they're there is because in Unit 4, you do have to know all of them. I'm trying to get you seeing them and kind of familiarizing yourself with them, but you don't need to come and memorize all of this stuff. And that especially goes for our exotoxins, but I want you to see how diverse they are because exotoxins are very dangerous. And so far we've said that they're made in the bacteria and released. And so that's why some are neurotoxins. When the bacteria releases that toxin in the host, it goes and affects the brain. Some of them go and affect the respiratory system. And you can see there are several ones that do that. Okay, some go and infect the gastrointestinal tract. So those toxins are able to do that because the bacteria makes them and then basically like throws them at the host. And it goes and circulates within the host and targets the brain or whatever. Okay, so that's a big difference with exotoxins. They're very, very diverse. Okay, so these are proteins, and that's really important because most of them are enzymes. Specifically, they have a job. Okay, and so because of this, I'm going to kind of go a little bit out of order. Because every enzyme has its own unique function, right? We learned that in Unit 2. No two enzymes do the same thing. There's no need for that redundancy. So every toxin, exotoxin, acts differently, right? That's what makes botulism botulism, and anthrax anthrax, and tetanus tetanus. So we do say that these are disease specific. Okay, because no two enzymes do the same thing. No two toxins do the same thing. See, now I have no idea what I have just done to my writing. Okay. So this is disease specific, okay, because the toxin is an enzyme that is going to have its own unique job. Now, because they are proteins, they are heat sensitive, right? We talked about in Unit 2 that proteins, when they get too hot, they will denature. That means they fall apart and they won't work. So because proteins can denature, it doesn't make sense for an exotoxin to cause a fever. Because if it caused a fever, it would essentially be destroying itself, right? And that doesn't make any sense. Pathogens aren't trying to wage war on themselves, okay? So they do not stimulate a fever because they don't want their strongest weapon to fall apart, okay? The other big thing is because these exotoxins are enzymes, they each have their own special signs and symptoms, their own problems that they cause. They are disease specific, but they also have a very low LD50. As I was saying a second ago, botulism, it takes the tiniest amount to kill something. Okay, anthrax also has a pretty low LD50. So again, they're very potent, they're very dangerous because they're, causing our, brain not to do what it's supposed to, or our digestive system to go cuckoo birds. Okay. So you'll definitely make sure you know the differences between endotoxins and exotoxins. All right, we're going to go ahead and go through number 4 a little bit quickly. But again, you have this now as a resource. You can come back and, as you're studying for the test, fill it out based on memory. And again, I'm sorry this isn't more interactive. I'm just trying to get through everything in an hour. All right, so determine if the following statements are true or false. Correct the false statements. All right, proteases lyse red blood cells. This one is going to be false because a protease, sounds like protein, right? So there are 2 ways you could make this correct. You could say proteases lyse or destroy proteins, or you could change this first word and it would be hemolysins, lyse red blood cells. Okay. Hemolysins, lyse red blood cells. So remember, hemolysins, proteases, catalase, coagulase, some of these that you've hopefully been learning about in lab unit 2, these are all exoenzymes. So these are just more little weapons that bacteria can possibly use, okay, to try to weaken the host and help spread better within that host. Okay, so that the protease, hemolysins, those are all examples of exoenzymes. All right, an incubatory carrier will eventually get sick. That one is going to be true, okay? And so remember when we talk about carriers, we have like a human reservoir, and so that's when we use the term carrier. So remember, asymptomatic carriers will never get sick. They're asymptomatic forever. Incubatory carriers are only not sick during that initial incubation period, but eventually they'll hit the other parts of the curve that we'll talk about soon, and they will get sick. Okay, so asymptomatics do not get sick. Incubatory carriers will eventually get sick. All right, strep throat is a zoonotic disease. That is not true. This passed person to person. Okay, so rabies would be an example of a zoonotic disease because you have to be bit by like a dog. Although typically it's not a dog. Actually, it's very uncommon for dogs to have rabies. They can, but typically it's more like raccoons and things like that. Okay, D, the higher the the higher the ID50 of an organism, the more virulent it is likely to be. So that's going to be false, okay? So you could say the lower the ID50, the more virulent it is, or you could have kept higher and made this one less. And there's normally multiple ways to revise these statements.
Is it possible for something to have a really low ID50 and a really high LD50?
No, those are always associated with each other. Because typically, well, no, not normally, because typically things that have a low LD50 means it can kill you. And there are a lot of pathogens that it would take an insane amount to actually kill you. Like it's not really probable that it would happen. And so normally, if something is that virulent that it can kill you, most of the time it doesn't take much to get in. So with science, like I don't like to ever say that'll never happen. So I'm sure there's an exception or two out there. But let me think of how to explain this. With infectious dose, you're basically trying to say how much will make you sick. But for organisms that have those exotoxins, which is typically what makes something lethal, a lot of them, their exotoxins are so potent that it doesn't take much. So most things, if they have an LD50, it doesn't take a whole lot to kill you. But we do still compare them along the spectrum. But a lot of those really dangerous ones also have a low ID50. So is it possible? Yes. It's just not very common.
All right.
Because a lot of them, as we'll learn in Unit 4, a lot of them are spread by endospores, which are those little very dormant forms of a bacteria. And so when you tend to come into contact with an endospore, like in the soil or something, you tend to come into contact with a lot of them at once. So even if it is like a higher ID50, you still tend to get it all at once.
Okay.
Okay, good question. All right, viruses use adhesins to bind target cells. This one is going to be false because adhesins are a bacterial structure. Okay, so adhesins are bacterial. Viruses use spike proteins to attach to their target cell. All right, so for F, if a disease is communicable, it is also contagious. This is false, okay? So remember, communicable means it can be spread. Contagious means it's spread really easily. And the reason this is false is because some diseases, like the ones I studied in grad school, which is Legionnaire's disease, it is communicable. Technically, no, sorry, that's not a good example. Let's use leprosy. Leprosy is communicable. It can spread person to person, but a common misconception. It actually takes a lot of like up close and personal, like breathing on each other kind of contact to get leprosy. People often think if you're close to somebody with leprosy that you're going to get it, and that's not true at all. So leprosy is communicable, but it is not contagious. Okay, strep throat is contagious, right? If somebody with strep throat coughs on you, like you're sick. That's just how it is. Okay, so a disease can be communicable and contagious, like strep throat. One can be communicable and not contagious, like leprosy. And actually, Legionnaire's disease, which is what I studied, is not communicable. It cannot spread from person to person. You can only get it if you inhale contaminated water droplets. So it's very hard to get, thank goodness, because it's a nasty disease. Okay, so it can be communicable and contagious, communicable, not contagious, or not communicable. All right, G, antigenic variation is an evasion strategy used by some bacteria and viruses. That is true. Remember, antigenic variation means that it's just changing up its surface structures. Because when it infects us, if its little antigen is a square, we're going to make antibodies that have that square shape to catch it. But by the time we make those square antibodies, if it now has round receptors, well, those initial antibodies aren't going to do us any good, right? Because now it's changed its shape. So then we make antibodies against the round antigen. Well, then it switches it up again. Now it has triangular antigens. So now the square antibodies are not doing us any good. The round antibodies aren't doing us any good. So now we make triangular antibodies. Now, eventually, like the bacteria or virus, whichever, it only has so many tricks up its sleeve. Eventually, it's going to go back to that square or round antigen, and those antibodies will get it, okay? But it's just like a delay tactic. It's trying to hide for a bit longer. Okay, so it's a very effective strategy that bacteria and viruses use. It's kind of like when human criminals go and change their appearance, right? They robbed the bank, they had glasses and a beard. Well, then they go and shave and wear contacts. It's a lot harder for the police to find them, right? But eventually, the police are now looking for a non-bearded, person. All right, last one. If a bacteria's preferred portal of entry is the respiratory system, it will most likely exit the body via the respiratory system. That is true. The preferred portal of entry and the exit portal are always related, okay? So if somebody coughed on my face, right, and it got into my respiratory system, and when I cough on somebody, I'm going to release it that way. Or if I sneeze on them, I can get them that way. If I eat contaminated food, that's the digestive system, then typically if people come into contact with like feces, right? Again, the end of the digestive system is how they can get sick. And when we get into food poisoning, a lot of times it's because people don't wash their hands after they go to the bathroom, right? Go. So the entry and exit are typically tied to the same system. And remember, the respiratory system is the most common portal of entry, right? Because we breathe all the time. We talk all the time. So we're constantly giving pathogens access to our nose and our mouth. All right, so I'm going to skip the number 5, sketch and label the graph showing the parts or the stages of a disease. And that's because I'm just going to put it on the screen instead. That's this. This is what we would be sketching out, okay? So make sure you're familiar with this because this is going to come into play as well in Unit 4. So let's just talk through it really quick. So incubation period is always what happens first. This is when you're infected, okay? Somebody has sneezed in your face. The bacteria, for example, are actively replicating in your body, but you don't know it. Okay, you do not feel any different. So this is infection. Okay. When you kind of wake up or it just kind of hits you all of a sudden, you're like, oh, my throat's a little scratchy, right? That's unusual, right? Now you're in a diseased state, but it's a vague symptom, right? Your throat's scratchy. At this point, you're like, is it allergies? Is it strep? Like, I don't know. You're kind of tired. You don't feel good. You feel off is how we normally describe it. That's prodromal. By the time you get to invasive, notice the number of pathogen is really high. Okay, so here you are full-blown sick. It hurts to swallow. Your throat is red. You've got those pus pockets. Okay, you're not going anywhere because you're miserable. Okay, that's the invasive phase. The point in time when you have the most number of pathogens that you're going to have, that's called acme. And at this point, the immune system kind of comes into play. It takes a while. You'll learn next week. It takes a little bit of time for our specific immunity, which involves like antibodies, to come into play. We try to let our body handle it like without the big guns at first, but as we know, they don't always get the job done. So about when we hit acme is when the antibodies and the big dogs start to come in. They start rounding up and eliminating those pathogens. So we start to feel better, okay? And then eventually we've gotten rid of most of the pathogens. You'll see there's still some here, but we're basically back to our normal self. Okay, so we're not really in a diseased state anymore. We have no more signs and symptoms. We're in the convalescent period. But notice, y'all, anytime that there are pathogen in our body, we are technically contagious. We can spread that pathogen. Okay, you may not have a ton, but they're still there. So if you go coughing and sneezing on people, they can technically get it too. Okay, so in all of these phases or stages or periods, you technically can pass or spread or transmit that pathogen. All right, so I'm gonna go ahead and wrap up there. Make sure, y'all, that you know all of these terms, okay? This is a huge unit, okay? I'm gonna see if I can try and find like an extra time next week to maybe do 2 reviews and a lab review. This 8 weeks are crazy. I give you a lot of credit for doing this. There's a lot going on. But just all of these terms, okay, make sure you're familiar with what they are, because on this exam, there's going to be a lot of scenarios. Okay, so we'll just stick with the one I've used over and over today. You know, my niece sneezed coughed in my face. The germs got in my mouth, my throat, and I eventually got strep throat. Well, that's a localized infection, right? Because they entered into my throat, they stayed in my throat, and eventually I cleared it. Okay. So that's what I mean by scenario. But if I tell you, you know, I don't know, somebody got the flu, right? And they got it because they inhaled in the virus, but then the flu can affect everything, right? It can make you, some people puke, you get aches and pains all over your body. So you would tell me that's more of a systemic, okay? So make sure you're really understanding what these terms mean and how they would look, because from here on out, it gets a little bit harder to just like memorize your way through. You need to be able to apply it to a situation. All right, let's go ahead and talk about our non-specific immunity real quick. So this flow chart is pretty much like the one that's in your non-specific immunity PowerPoint, okay? You're gonna want to be able to fill this out on your own. I'm gonna go through part of it real quick because I want us to really talk about the processes more so than this chart. But when we do the review session next week, we are going to come back to this because this is what we're talking about next week, right? And don't let the very few number of boxes fool you. There's a lot that goes into these little boxes. Honestly, it probably is part of it, yeah. So I'm not going to give you all the answers, but I want to kind of get you going. You mean this week's week 5 or week 4? Which one are you talking about, Paula? The week 4 activity is over non-specific immunity. Yes. I honestly don't remember what I put on that, if we're being completely honest. I remember part of it. I remember the flow chart. Now I'm about to go look it up. Give me 1 second. I don't remember what I put for, non-specific was oh, no, you're fine. Non-specific, it was all those processes, yeah, where you had to make like a common term, things like that. So this was from non-specific immunity. So we're going to talk about this part next week. Currently, right, we're over here with our non-specific immunity. Okay, and from there we have our first lines of defense. And then we're going to have our second lines of defense. And for a lot of people, the first lines of defense, oh, okay, no problem, Paul, no problem. Okay, so with our non-specific immunity, okay, here's what you really need to remember at this point. These are going to be cells. They all involve cells, which lead to processes that are meant to prevent things from actively replicating in your body. It's trying to limit the spread of a pathogen. So our first line of defense, the goal is to keep things out. Okay, so I'm going to limit how much I write because I'm using a mouse and it's hard. Keep things out of the body. So this is going to be things like physical barriers, right? Like our skin. Our skin, tightly packed cells, very dry. We shed that upper layer. We're meant to keep things that get on our skin out. Okay, and then we have those chemical factors, things like lysozyme, which you find in our tears, in our saliva. And so that when anything gets into our eyes, it gets into our mouth, the lysozyme, some of those chemical molecules, are breaking down those bacteria. So they're little molecules that, again, try to kill things as soon as they touch us so they can't replicate in our body. Okay. And then we also have those mechanical processes, coughing, sneezing. When you blink and your eyes water, again, those are mechanical processes meant to flush things from our system. Like, okay, cool. I just inhaled, I don't know, the flu virus when you sneezed in my presence. Well, my body's going to be like, no, thank you. We don't want this. And it triggers things to sneeze to try to also clear your nasal passages. Okay. Now, we can also put in here our normal microbiota. I'm going to put that as NM, the normal microbiota, because they are part of our body. You know what I mean? Like, it's not an organ, obviously, but they're in there and they're helping keep pathogen out. But this is just one of those topics that some researchers like debate, that they're not part of our body like when we're born, So I consider it part of our first line of defense because the bacteria that lives in our body, they don't want pathogens to come in and take over, right? We don't want people to rob our house. We're the house for these little bacteria. So normal microbiota can also be considered, in my opinion, a first line of defense. So make sure, again, examples. On a quiz or an exam, I might say, coughing is an example of what? And you would say it's an example of our first line of defense. Maybe it'd be more specific. Coughing is a mechanical component of our first line of defense. So make sure you know examples. If I were you, I would really build this out. I love flowcharts personally. So if they work for you, I would extend this and give more examples. All right. So here, if you kind of scroll down on the handout, is where we're going to talk about the different components of our second line of defense. So everything we put in this table would just go right here. And again, it's already on the PowerPoint for you. But when you're studying, I highly recommend y'all that you be able to fill this out without hesitation. Okay. And especially be able to build them out. Okay. Yeah. Maybe you're going to put phagocytosis right here, but then can you draw a line and tell me what that is? So let's go ahead and do that. I know we're only down to like 9 minutes, but we got this. All right, so what I'm going to recommend you do is I'm not going to speak for about 30 to 45 seconds. I'm going to be typing in our 8 components of our second line of defense. I want you from memory to do the same thing. Okay, so just fill out this left column with the cells and processes that make up our second line of defense, because I promise you want to know these. So ignore my screen and write this on your own. All right, once you've listed as many as you can from memory, go ahead and fill in whatever you're missing from my list. And we're going to go ahead and start, because we're down to 7 minutes and 8 things to talk about. All right, so my favorite one, first one, phagocytosis. Love this. I think it's so snazzy. The goal is to find, I'm going to say capture or catch. I'm going to really have to let go of spelling right now. Find, capture, and eliminate pathogen. Okay. Another way I tend to say this is ingest and digest pathogen. Okay. So how does it do this? Okay. And you have a slide in your PowerPoint that has all the steps spelled out. Okay. So I'm not going to just do the same thing. But with this any of these processes in these cells, you need to know those key terms associated with it, hence what you did in your week 4 activity. You need to know the terms and kind of what does it mean? Why is it important? Okay. So for phagocytosis, in order to eliminate a pathogen, like to kill it, you have to find it, right? And so that's what chemotaxis is. Think about when people are looking for a missing person, right? They take some of their clothes and they give it to like a hound dog or whatever. The dog sniffs it, right? The clothes, and then they sniff the environment and they follow the scent. That's what chemotaxis is, okay? The phagocytic cells, our immune cells, are literally sniffing out and hunting down that pathogen, okay? When they find it, they grab hold of it, okay? Eventually, they're going to do endocytosis, which is the ingesting part. Okay, that's more of a common term, is ingesting. They take it in through endocytosis, and they kind of keep it. They put it in there, its own little jail, its own little vacuole, right? That little jail cell, that vacuole, is called the phagosome. So I'm going to put in parentheses kind of the common terms. But you guys always speak micro, okay? On exams, on unknowns, always speak micro. So they put it in the jail. It's hanging out there in the phagosome. But now we need to get rid of the pathogen, right? That's where the lysosome, all those hydrolytic enzymes come into play. That's what breaks things down and quote unquote kills, like the bacteria. So I think of it as like the executioner, okay? So when the executioner has come, that's when the phagolysosome has formed. It's the pathogen plus those hydrolytic enzymes, okay? So this is my executioner. And that is also going to be when the digestion happens. This is when the bacteria, for instance, is being chopped into bits, okay? Once that's done, right, you got to kind of clean up the mess. All the leftover debris is the residual body, okay? So everything that didn't get digested, kind of like the trash, it's just going to get taken out and eliminated, okay? So those are just some key terms. So they have to find the pathogen, they grab hold of it, they ingest it, they digest it, and then they get rid of the waste. Do we have any questions about that? I know I'm at this point kind of going quick. If you do have questions, please jump in. Okay. Interferons, people call this the Paul Revere of the cell. I think of it more as like an alarm system. The purpose is to warn the body that we've been infected. We are under attack, okay? So in the PowerPoints, there are like 3 outcomes of interferons. You're gonna want to know what that is. But essentially what's happening is an infected cell is going to release the interferons as a warning that they are being attacked. Again, you don't have to write all this that I'm typing. But think of it as like an alarm system on our house, right? We set our alarm before we go to bed. So if somebody tries to break in, that alarm is going to notify us first, right? Hey, somebody's trying to get in. You need to arm yourself or hide or whatever the case may be, right? But it also sends a signal to neighboring houses like, hey, yo, y'all need to lock down. There's a problem, okay? So these interferons are kind of warning other people, but also saying, hey, if you're infected, you gotta go from a cell perspective, okay? So these are made by infected cells, okay? And they're warning uninfected cells so that those cells can arm themselves, okay? And so those uninfected cells tend to make AVPs, antiviral proteins, which are their little military, okay? They get all their little weapons and they kind of stand guard and they're like, all right, virus, if you try to get in here, we're gonna get you, okay? So again, infected cells make and release interferons. All the surrounding cells receive that interferon, and then depending on their status, they respond. If they're like, ooh, there's an infected cell, they receive that message and they go, ooh, am I infected? And there's ways of kind of like assessing what's happening in the cell. If they are infected, those cells undergo apoptosis because they're trying to essentially take one for the team. By having those cells implode, the virus cannot get out and infect other cells. Okay? If the cells kind of check themselves and they're fine, then they're going to make those AVPs, get their little military ready, and they stand guard. And if a virus tries to get in, they break it down. They also go and kind of call in the big dogs. They alert our specific immunity that you'll learn next week. Hey, we're under attack. We may need you. And to get ready, we're not sure how bad it is, but you might be needed. Okay? All right, inflammation. This is meant to quarantine pathogen. Okay, so in the picture, it's like this horrible like nail or nail file that's penetrated the skin. Stuff is going to be on that nail, for instance, right? That can get into your body. We don't want any pathogen that has now gotten in to spread. So with inflammation, one of those key indicators is swelling. We're trying to limit the pathogen to that one area. Okay, so remember for this, one of the first things that's going to happen is mast cells release histamine. That's going to be what actually triggers the swelling and the redness and all of that. When histamine gets released, it's going to cause vasodilation, which means the vessels dilate, they get larger. This allows, imagine these are little arrows, guys. This is going to allow the white blood cells, those phagocytic cells, to march to where the infection is at. That process is called margination. This is those phagocytic cells, those immune cells, marching to the site of infection. Okay? Once they get there, they've got to squeeze through the vessels in order to actually access the pathogen and round them up. That is called diapedesis. Okay, so imagine like getting off the elevator. They're at the site of infection. And then from there, they do phagocytosis, where they round up and eliminate the pathogen. And it's that vasodilation, y'all, that really causes the redness, the swelling, kind of the pain associated with inflammation. Because y'all see lots of terminology here, right? So make sure you're familiar with that. Now, I know it's 11. I'm going to keep going just to finish this so that you'll have it recorded. If you need to go, I completely understand, and I apologize for running late. All right, so next up are the nets. The N stands for neutrophil. That's the type of cell, okay? But it's the process of forming a net and the process of eliminating pathogen. That's why it's called process in that first column. So these are neutrophils, and what they essentially do is they create a net, like a big old fishing net, but they make a net out of their own DNA and enzymes that they literally release from their cell to capture the bacteria. Okay, they release the net and it covers and eliminates the pathogen. Now, y'all, this is cell suicide because the net is their DNA and they throw it from the cell. They literally punch a hole in themselves to release that net. So these neutrophils die in the process, but it will literally trap the pathogen, and then these little enzymes go and break it down. Okay? So the neutrophil does die in the process, but it's a very effective way of trapping and, in this case, killing the bacteria. All right, so pyrexia. Oh, sorry, I forgot the purpose here. Trap and eliminate. I like to use the word eliminate rather than kill, because if it's, you know, a virus or something, typically these are for bacteria and fungus. But I just try to use, you know, more neutral terms just in case. All right, so pyrexia, we're cranking up the heat, right, to try to kill or eliminate the pathogen. And so some key words with pyrexia is, remember, an exogenous pyrogen, okay, is going to be detected by an endogenous pyrogen. Okay, that's going to cause the I'm going to talk through this a little bit more once I type it. Hypothalamus will reset. Typing are not on the same page today. Hypothalamus will reset to a higher temperature, and then the body will respond to increase the temperature. I'll talk about that here a little bit more. Okay, so pyrogens are going to be kind of your key science terms for this. And pyrogen sounds like pyrexia, right? So remember, exo and endo is what you see here. Endo means within, exo means outside of, okay? So an exogenous pyrogen is a pyrogen that does not come from the body. Our body is going to recognize it as foreign, and then our own molecules are going to go and alert the hypothalamus that we need to crank up the temperature, okay? Once the hypothalamus knows that we need to increase the temperature, our body is going to respond, okay? And there's a slide that talks about kind of 3 of those big processes that our body does to crank up the heat. It does vasoconstriction. It constricts those blood vessels because we want to keep all the heat in, right? We tend to lose body heat at the surface of the skin, right? That's why like our digits and our nose and our feet tend to be more cold because we're losing a lot of our heat at those places. So by constricting the vessels, we're minimizing how much heat we lose. We're going to start shivering and getting the chills because as our muscles move faster, they're generating heat. The faster things move, the hotter it gets. So again, this is our body's physiological response to try to crank up the heat. And then some of our like thyroid hormones are also going to increase metabolism. Again, that's with all chemical reactions in the body, they naturally give off heat as a byproduct. Okay, so increasing our metabolic rate, shivering, and vasoconstriction are 3 responses the body does in order to reset our body to a higher degree. Okay, any questions so far? I know I am going pretty fast, and I'm sorry about that. Unit 3, honestly, is just a doozy. There's so much here. Okay, antimicrobial peptides. This one is a bit easier. As far as we know right now, it tends to target living things. And so I am going to put kill here, kill the pathogen. And it does so by poking holes in the plasma membrane. And really, this one's relatively straightforward, okay? This is a short chain of amino acids, hence peptide, okay? That fold, okay? They fold together. There's a picture on your slides. And when it's folded, this peptide is amphipathic, a word you've heard before. Remember, that means part of it is hydrophobic, and the other part is hydrophilic, okay? So if you go back and you watch that video or you look at that picture, the hydrophobic part of this peptide is compatible with the plasma membrane, right? It's a hydrophobic barrier. So the hydrophobic part of this peptide inserts into the hydrophobic part of the membrane. That's going to essentially allow this peptide to be poking holes into that membrane. The membrane's going to leak. The bacteria, for instance, will die. Okay, so this molecule inserts, and there's lots of these peptides, okay? So they all insert into the membrane, causing it to leak. And the organism, like the bacteria in this example, will die. All right, 2 more. I'm going to be quick. I know I say that all the time. The natural killer cells, that's what NK stands for. This is going to recognize foreign and infected cells and then ultimately destroy them. Okay, so this is where the NK cells are going to determine or assess the number of MHC1 on a cell. Okay, so real quick, remember MHC1 is found on all of our cells. So if the cell belongs to us, it should be in the body, it's going to have MHC1 on them. So think of the natural killer cells as kind of like checking to make sure you belong. Okay, so here's my natural killer cell. If you can see my little video on the bottom left. Natural killer cell is cruising around the body. When it finds any other cell, it stops it. It's like, hey, you have MHC1? Yeah, okay, cool. You belong. Keep going. So if it comes across a pathogen that does not have MHC1 because it doesn't belong to us, it shouldn't be in there, it's going to destroy it. And we'll talk about how in just a second. So that's seeing whether or not something has MHC1. That's one way to detect pathogen, because pathogen is not going to have MHC1. The cells that do belong will. But notice I also said that it assesses the number of MHC1. So healthy self cells have a set number of MHC1. I'm just going to make up this number. Okay, so pretend all normal human cells have 100 MHC1 on them. It turns out when our cells are either, we have cancer, like there's cancer cells, or virally infected cells, right? They're sick. That number of MHC1 decreases drastically, sometimes to where there's none left, okay? And so when a natural killer cell encounters a sick self cell, it might see MHC1. So it's kind of really asking two questions. Do you have MHC1? Yes or no? If no, it destroys them. But if it says, yes, you do have MHC1, the follow-up question is, how many do you have? Do you have that 100 that you should? Oh, you do? Cool. Go ahead. Go about your business. Oh, wait, you only have 10? Ooh, that's not normal. You must be infected. I need to destroy you. Okay? So again, does it have MHC1? No, destroy it. If it does have MHC1, you ask, how many do you have? Do you have the correct number? If not, you get destroyed. Okay? So if the cell that it comes into contact with does not have MHC1 or it does not have enough MHC1, it is going to go ahead and do apoptosis. Okay, that's the end result. Where'd my little mouse go? Here it is. Apoptosis, which is programmed cell death. It causes that infected cell to implode specifically. Okay. So it does that. If you look back at your slide, there are certain little proteins that the NK cell has called perforin and proteases. The perforin is going to poke a hole in the infected cell, and then it's going to release proteases to go and kill all the proteins in there and cause the cell to die. Okay, so you might want to know granzyme and proteases are what's responsible for causing that apoptosis. It's really what kills that infected cell or that pathogen. Okay, last but not least, the complement system. This does a lot, okay? So the purpose is kind of hard because there's so many things that it does. So it's meant to we'll say identify, and I guess identify and eliminate is fine. Eliminate the pathogen. Remember, there are 3 different things that can activate this system, okay? But essentially what happens is once it has identified that there is a pathogen in the body, which it can do 3 different ways, its goal is to kind of get rid of it or to help the specific immunity do its job. So a lot of people think non-specific and specific immunity are like 2 things, they never interact. Well, that's not true. They're parts of our overall immune system, so they are going to be working together as well, okay? So there are really, you want to know the 3 outcomes of the complement system, okay? So one is going to be a process called opsonization, which is where it's going to cover the pathogen to enhance phagocytosis. Okay. It's going to cover the pathogen in little complement proteins, which is going to allow our phagocytic cells to be like, oh, you don't belong here. And it's going to grab that pathogen and it's going to digest it. Okay, so opsonization covers the pathogen in complement proteins, which signals the phagocytic cells like, hey, this thing needs to go. So then the phagocytic cell will come and phagocytose it. The other thing it's going to do is it's going to trigger inflammation. Because again, notice this system is identifying the problem. It says, whoa, guys, look, I found this E. coli cell. We need to quarantine it. We don't want this bad boy to spread, okay? So it will trigger inflammation. So again, notice that these components, they do work together. They're not their own little lone rangers, okay? They do work together. It causes inflammation, again, to try to prevent the pathogen from spreading. And then the other is cytolysis, okay, via formation of the MAC complex, the membrane attack complex. And basically, this is where it's going to go and poke holes in the pathogen's membrane and try to eliminate it, okay? So the complement system is really effective because it doesn't just do one thing. It does lots of things. So it really can help us round up and find and catch and eliminate a lot of pathogens by doing these three outcomes, okay? Because, and we'll talk more about opsonization next week, but remember, some pathogens, a lot of them have a capsule, that sticky layer, right? That makes it hard for phagocytic cells to grab hold of. And so one way we fight back against that is by cutting that sticky layer with complement proteins, by opsonization. Because now that pathogen is not sticky. Now it's covered in little proteins. So the phagocytic cell can use those proteins like handlebars. It can grab hold of it so it can ingest it and digest it, okay? So it helps us actually better eliminate those pathogens. It traps them in place so that the phagocytic cells can get to them. And it also is trying to eliminate those pathogens directly by poking holes in it as well. Okay, so complement system is kind of a beast in terms of how good it is at trying to clear pathogen. All right, y'all. Again, I'm very sorry that this went so over. Hopefully it helped. If you have questions as you're studying and working on things, please let me know. I was in a hiring committee from 8 to 5 yesterday, so I was not very accessible. So if anybody did reach out, I apologize if you haven't heard from me yet. I will be going through my emails and stuff soon. As always, Remind app is a great way to get a hold of me. And don't forget, if you haven't already, you need to respond to your peers' discussion posts. All right, y'all, you're very welcome. I hope you have a fabulous day. Happy studying. Let me know if you need anything. Bye, y'all.